Hi, my name is Ira Wahyuni binti Abdul Hadi with matrix number 99162. Today I will be presenting the stage and modern advancements of drug discovery and development. Drug discovery is a complex process involving the identification of therapeutic chemical for disease treatment and management. It involves several stages to ensure the medicine is safe, effective, and approved by regulatory requirements. The first stage is target identification. Here, we identify the biological origins of a disease and potential target for intervention using methods like data mining, using bioinformatics, and functional screening. This contributes to the overall process by giving us a specific biological target to aim our drug at. Next is target validation, where we demonstrate the functional role of the identified target in the disease by reproducing experiments. This test ensures that hitting this target will actually help manage the disease, preventing worse effect later on. Once the target is validated, we move to lead identification. This involves finding a chemical compound that interacts with our target, evaluating its synthetic feasibility and preliminary toxicity to give us an initial drug candidate. Then comes lead optimization. In this test, we alter the chemical structure of our lead compound to improve its stability, bioavailability, efficacy, and reduce toxicity. This step is vital as it transforms a raw chemical into a viable, optimized preclinical candidate. The next stage is product characterization, where we define the optimized molecule physical properties such as its shape, size, strength, and biological activity. This knowledge directly supports formulation and development. Here, we characterize the active ingredient to create a bioavailable, stable, and optimally dosed formulation. This ensures the drug can actually be delivered into the human body effectively. Before human testing, we must conduct preclinical research. This involves in vitro and in vivo pharmacological and toxicological studies to evaluate the drug's safety and efficacy in animal models. If the drug proves safe, we compile all this data into an investigational new drug application (IND). Approval of the IND is the mandatory gateway to begin clinical trials. Clinical research is conducted in human volunteers across distinct species. Phase 0 involves very small doses to gather initial pharmacokinetic data. Phase 1 focuses on safety and doses in a small groups of volunteers. Phase 2 tests the drug efficacy and side effects on a large group of patients. Phase 3 involves large-scale monitoring for efficacy and adverse reactions. Finally, phase 4 is post-market drug safety monitoring. Each phase acts as a strict filter to protect human health. If phase 3 is successful, the developers submit a new drug application (NDA). This includes all previous data and proposed labeling to prove the drug is safe and effective. The FDA reviews this and, if successful, grants final approval so the drug can enter the market. As highlighted in the article by Nicolaou, modern advancements have heavily influenced this pipeline. For example, computational methods now allow us to predict a drug's ADMET properties, which are absorption, distribution, metabolism, accretion, and toxicity before synthesis. Another major advancement is polypharmacology, where drugs are intentionally designed to interact with multiple biological targets simultaneously. An excellent example of this is the breakthrough cancer drug imatinib or Gleevec, which target multiple kinases to effectively treat leukemia. Next is structural motif by introducing saturated and chiral motifs alongside halogen and halogen-containing residue to improve pharmacological properties. Lastly, we see a renewed reliance on natural products as primary lead because their complex 3D structure have evolved alongside biological receptors, making them naturally potent and selective.